Cytoplasm. Because only bacteria doesn't have nuclear membrane and has nucleoid. Yes, Mr. Hussein Muhammad, there is no nuclear membrane, so it is 100% bacteria. There is no nuclear membrane. Cytoplasm is another vital part. It is very important, cytoplasm. Cytoplasm contain water. All catabolism and anabolism have so cytoplasm cell, we say pilus. Pilus. And elongate, hollow appendage. Elongate, hollow appendage used in transfer of DNA to the other cells. Which other cells? He tried to make his friends more powerful. So some parts of DNA from this hollow appendage can be transferred to the neighbor bacteria. The neighbor bacteria. So transferred the power, because power of the persons and the power of the groups, both of them are important. Bacteria understand only personal power is not sufficient. Every person should be powerful, and every bacteria should make the weak bacteria powerful too. Yes, in previous section I explained that prokaryotic cells, which parts they have and which parts they don't have. Completely last section we explained that bacteria doesn't have the Golgi apparatus, they don't have the mitochondria, they don't have the reticulum endoplasmic, but this is the star one, ribosome are present. How made ribosome? Ribosome production. Ribosome has two parts, protein and nucleic acid. And each part has essential enzymes. So, according to the chromosome, some enzymes expressed by gene expression, some going to the cytoplasm for protein synthesis, some go for nucleic acid synthesis, and in combination of RNA molecules and protein molecules, ribosome comes to the existence. And the ribosome of bacteria is different with ribosome of the other organisms. Okay, so this is a hollow appendage, has two functions. One function, transfer the power. How? Transfer the DNA from one bacterium to the other bacterium. What is the other capability? Cell adhesion. They can attach to the surface, they can attach to the tissue, they can attach to each other by this part. What is its name? Pilus. What is the attachment for? For example, suppose one bacteria want to attach to the intestine cells. So should attach and then produce enzyme and destroy it. Attachment is necessary to have a good position for action. To have a good position, attachment. In bacteria, adhesion and attachment is very important. You suppose there is a palm tree. How can I get the fruits? the palms. I should go up, attach, and then catch the fruits. Without attachment, it is not possible to get the fruits. It's not possible to get the palms. So attachment for bacteria is very, very, very important to the place which they want to do something. Another place, as you see, there are thinner, shorter appendages. We say fimbria. Fimbria. They are very fine, hair-like bristles from the cell surface that help in adhesion. What is the usage of this fimbria? Adhesion. I told you adhesion is very important. Adhesion is very important. Adhesion to the other cells, adhesion to the surface, adhesion to the host, to the cells, very, very is important. So fimbria in one way is look like pilus for attachment. In one way is different. Pilus can transfer DNA, but fimbria can not. Outside the cell membrane, there is a layer like a pocket, like a coat. We say it is cell wall, like plants. Plant cells have cell walls. Okay, but composition of cell wall of bacteria is completely different with cell wall of plants, fungi, and the others. So bacteria have cell wall. What is the most usage of the cell wall? Mostly bacteria absorb food from outside. Mostly bacteria absorb food from outside, and inside is more concentrated than outside. Water prefer to go inside according to the gradient of material. Cell wall don't allows to break the cytoplasmic membrane to make the bacteria suppressed, to not allowing bacteria to be bigger and then erupting, not allowing. So mostly bacteria are in the place inside are more concentrated, outside is less concentrated. Water comes inside. Cell wall stand against the pressure of water. A semi-rigid casing that provides structural support. It's supporting and shape of the cell. If we see one bacteria is elongate, is according how these blocks of cell wall are together. If we say they are spherical, it is according to the combination of the cell wall. So when we go and observe by microscope a bacterium, any shape we see, it is according to the combination of the cell wall. Inclusion. Bacteria get food from outside as much as possible. Bacteria get food from outside as much as possible. Okay, I explain it. If it is so, we return back. There is no problem. Thank you, dear student Riaz. Yes, of course, Mr. Murtaza. Of course it is. The name of transfer of DNA from bacteria to the other bacteria is conjugation. So we are introductory phases. We are, as much as we go straight, we complete the explanations. Okay. So when we go to shopping for food, we don't get food as much as for one meal. We get more rice, we get more meat, we get for a week, for a month, for a few days. Bacteria, as much as they can, absorb food from the outside. A part needed for the moment living, and the extra is used for storage. This storage, we say inclusion or granule, inclusion or granule, stored nutrients such as fat, phosphate, glycogen, deposited in a dense crystals and particles that can be tapped into when needed. Whenever it is needed, for example, after lunch, after 1 hour, 2 hours, whenever needed, you go through the refrigerator, you go through the kitchen, you go through the stored food. No, pilus is not used for reproduction. Reproduction means the chromosome will be double. It is inside the cell happening, and the DNA which transfer is not the chromosome. We have small DNA molecules, different small DNA molecules with the name of plasmid. Plasmid. So we have two DNA, two types of DNA in the bacteria. One type is the chromosome, like the others. Okay? So chromosome finished. But unlike the other organisms which we knew before, we have one or some small DNA with the name of plasmids with extra powerfulity. For example, plasmid for antibiotic resistance, plasmid for metal resistance. These small molecules can be transferred by pilus from one bacteria with this power to the other bacteria without this power. So plasmid or small DNA molecules, not chromosome, small DNA molecules with the name of plasmid, we say plasmid extra chromosomal DNA. What is the plasmid? Extra chromosomal DNA. This plasmids can be transferred from one bacteria to the other bacteria of the same group, because bacteria They are very wise. They know personal power and group power with each other is helpful. So easily they transfer their powers to their friends. Okay? Did you get the point? So cell wall, cell membrane, ribosomes, flagellum. The other appendage, as you see, very bigger, very elongated than pilus. Something like pilus, but elongate, filamentous, and with rotation. It is flagellum. Flagellum. Specialized appendage attached to the cell by a basal body. This is basal body. It is at the base of the flagellum that holds the longer rotating filament. So it has two parts, flagellum, basal body, and the filament. The movement of the pilus by this rotation, bacteria go straight the cell forward and provides motility. So what is the flagellum for? It is for motion, for bacterial movement. I explained mesosome, I explained pilus, I explained chromosome, and the last one is the glycocalyx. The glycocalyx. The external layer of some bacteria, the most external part of the bacteria, of course in some bacteria, is the glycocalyx. The other name is capsule. Glycocalyx or capsule. Where is it? A coating or layer of molecules external to the cell wall. So first envelope outside the cell membrane is cell wall. The second envelope is the glycocalyx or capsule. It serves protective, adhesive, and receptor function. Protective, how capsule, this green parts, the outer layer, the most outer layer of the bacteria, capsule or glycocalyx, how it gives protection to bacteria. It is helpful against white blood cells like macrophages. You see, when bacteria comes into our body, white blood cells and macrophages go through the bacteria, go towards to the bacteria, and they want to eat them. By some temporary months, they phagocytosis. I'm writing for you. Phagocytosis. So white blood cells and macrophages wants to phagocytosis the bacteria. Why? Their temporary months. They want to swallow the bacteria. But when bacteria make itself bigger than the months, they cannot eat the bacteria. For example, if I have a pizza in a plate, I can't eat it directly because it is bigger than my mouth. It should be cut. So bacteria make itself bigger than the place which white blood cell or macrophage want to swallow it. So it cannot. So it is a protective mechanism, protective of bacteria in front of the white blood cells. Okay, I know that some persons has internet problem, but what can I do? There is some suggestion. One suggestion is that you are right now 19 good students. With each other, write by your native language or English complete course, complete section, and cooperate each other, send to each other. And another thing I can do for you, if one page you cannot understand or you didn't have the voice, ask me later in the WhatsApp. Of course, anytime one or two or some people doesn't have the internet or good connection. So if many times we stop, never we can finish the course. Some of students, if cannot, come to the class, because this class last week, if you remember, I didn't have the user and password. First should check the time. Maybe some of them, their time is 1 o'clock, not 3:45. So first, please them check the time of their class. 100%, some of them, they are from previous class, so this class don't allow them to join us. And if it is not so, in WhatsApp, send me their name and family. So, and what you see, the notice on the screen by a screenshot, send me. I give it to the director to check and solve the problem. But by myself, I cannot do anything right now for them. If you get the point, please explain them and send them message. And if anything else after the class in group WhatsApp, we can discuss. How can we get the match? I don't get the point. What do you mean? Okay. So we understand that some bacteria have glycocalyx, some have less, and some doesn't have it. Those bacteria which have glycocalyx, they are more powerful in front of the phagocytosis by white blood cells. So it is a protective mechanism for bacteria. Adhesive, sometimes two or more bacteria with glycocalyx attach to each other and will be bigger and bigger and make a colony. So it enhances their powerfulity. And they can attach with glycocalyx and capsule to the surface, to the cells, to the place they want to attack. But on the other hand, All these parts are antigens. What is the antigen? Whatever can stimulate the antibody is antigen. All the parts of bacteria, all the parts, all the items are antigens. Do you think external antigens mostly induce antibody or internal antibody, internal antigens? What is your idea? External antigens or internal antigens? Anybody responded? Of course, absolutely. Very good. Mr. Hussein Sabah. External antigens. For example, when we go to the airport, the police officers, external appearance, our appearance is more important, or internal, like kidneys, like our heart, like our, no. What reflects to the outside and induce outside is the external. So the outermost part of a bacteria is the glycocalyx capsule and cell wall, and their antigen very, very good induced antibody production. So it is the receptor for antibodies, receptor for viruses. From previous slide, I don't think there is any important. Here, I think I explained, yes, this slide number 8, one person asked me 8 and 9. This table was discussed. Mr. Hussein Muhammad, have you been in the last section? Did I teach this slide number 8? Nobody knows? Okay. The question was slide number 8, this page. Did I teach last Tuesday or not? This picture is about the difference between eukaryotes and prokaryotes. The differences between eukaryotes and prokaryotes. Okay. So you can see some characteristics, eukaryotes and prokaryotes. Major groups, eukaryotes, algae, fungi, protozoa, plants, animals. Prokaryotes, bacteria. Size approximate more than 5 micrometer, plus bacteria or prokaryotes 0.5 to 3 micrometer. Nuclear structure, eukaryotes, yes, they have classic membrane, they have nuclear membrane. Prokaryotes, there is no nuclear membrane. Chromosome, eukaryotes, multiple and diploid. Okay. But in prokaryotes, one single chromosome, closed circular DNA chromosome. Cytoplasmic structure, mitochondria, in most eukaryotes present, not all, but in most of them. In bacteria, there is no mitochondria. Golgi apparatus or Golgi bodies present in eukaryotes. In prokaryotes, absent. Endoplasmic reticulum present in eukaryotes, absent in bacteria. Ribosome, ATS in eukaryotes, 70S in bacteria. Cytoplasmic membranes contain sterols, doesn't contain sterol in prokaryotes. Cell wall, eukaryotes absent. Some fungi like chitin, and some photosynthetic, maybe the others like cellulose. But in bacteria, present cell wall, but completely different with cell wall of eukaryotes. Reproduction, asexual, and in most, not only asexual, but also sexual is present. Reproduction in bacteria only asexual. Binary fission, I explain you in the next slide. Movement in eukaryotic with flagellum, if present, that flagellum is more complex, but flagellum of bacteria is very simple. Respiration, okay. Some students cannot enter the lecture. What is the problem? Please solve it. See if first step, user and password. If it is okay, comes inside the room. Now in the room, they should check the time of their class. You are in 4 groups. 1 group, 2 groups in the morning, and 2 groups in the afternoon. 2 groups in the afternoon, 1 group starts 1 o'clock to 3:45, and 1 groups now are ongoing. They should see what is the time of their lectures. If all the things are okay, they can come inside the room and their times is right now. So only I can get their names and send the director and asking him to solve the problem, the most probable for next section, because now the persons in the now only presentation is ongoing. The university now is closed. Nobody is answering right now, but I get a number from them for the possible problems in the future. Right now, unfortunately, I can't do anything. Okay, WhatsApp group was created by the students, and you can get the link from the other students, or from different ways you can join. If it is necessary, I do it. You can send me one WhatsApp to me so I can help you. Okay, respiration. What is the respiration meaning? It's very simple. See, why all organisms like us, like bacteria, need food? For what? For energy. They want to walk, they want to movement, our hearts wants to work, our brain wants to work, so we need energy, so we need food. What is the respiration? Respiration is And food like sugars, like starch, like corns, like galactose, maltose, glucose. What is the respiration? Breaking down the sugars, the organic matters, and produce energy. Where is the site for this respiration? In eukaryotes, mostly, but not all, mostly via mitochondria. But bacteria doesn't have mitochondria. Some bacteria have respiration, but their respiration is done by cytoplasmic membrane. Cytoplasmic membrane. So bacteria doesn't have mitochondria. Bacteria doesn't have mitochondria. And if they have respiration, their respiration is done in the cytoplasmic membrane. Today, Mr. Murtaza Sajil, after half an hour, we will discuss the absorption of food, oxygen, water, and the other things. So we will go with each other. It is a few minutes waiting, I will discuss you. Okay, this is slide number 8, finished. This is slide number 9. There are many certain structure, genetic, metabolic characteristics. And on the other hand, important biochemical differences exist among many species of bacteria. So they have many similarities. And they have many differences. Their similarities and their differences are according their structure, according their genetics, according their metabolic characteristics, according their biochemical behaviors. These differences permit bacteria to live in many different environments. Because of this diversity, we can see bacteria all over the world. We can see bacteria under the sea. We can see them under the oceans. We can see above the mountains. We can see at home. We can see at the street. Why? Because they have too much diversity. They have too much diversity. If you receive photo by delay, it is according to the internet. So it is a repetitive sentence we are hearing. It is according to the internet. Parts of the prokaryotic cells. On the photo, I told you that prokaryotes doesn't have true nucleus. And in bacteria, we say nucleoid because they don't have nuclear membrane. And on the figure I showed you, I explained you that bacteria have single circular molecule of DNA.
You are now muted.
Without mitochondria in eukaryotes, respiration not completed. It is a tool. It is a tool for completion the respiration. What is the respiration? Energy production from food. Just very simple. Okay, the exterior of the cell usually has glycocalyx, flagellum, fimbria, and pili. I showed you. Outside the cell is glycocalyx, flagellum, fimbria, and pili. One time again, see the photo. Where is the inside and where is the outside of the bacteria? Who tell me? From which border we say it is inside, and from which border we say it is outside? Who answer me? I know most of you answered. If one person message received late, it is because of internet. From which border is inside and from which border is outside? See, suppose one person is standing in front of you, okay? Ask you from which part of me is inside of the body and from which part. You say this coat is outside layer. This shirt is outside layer, okay? And a skin, after a skin, you go inside, it is inside from a skin. Now, cytoplasmic membrane is the border of inside and outside the bacteria. So we have internal organelles and external organelles of bacteria. DNA is external or internal? DNA. Is DNA internal or external? Very good, Mr. Hussein Sabah. Ribosome, internal or external? Very good. Glycocalyx? Glycocalyx. Very good. External. So our boundary of internal and external part is the cytoplasmic membrane. What? There are some envelope, there are some appendages outside the cell, and there are some appendages and some parts inside the cell, inside and outside the cell. So cell wall is outside layer, cell wall outside layer, glycocalyx outside layer, pili outside appendage, flagellum outside appendage. Okay, very good. Now we have one word archaea. Till now, we had two words, eukaryote, prokaryote. Prokaryote was bacteria, eukaryote and prokaryote. But we have one word with the name of archaea. What is the meaning of external same out? In the figure, whatever you say outside cell membrane is outside layer, like cell wall, like glycocalyx. Whatever is outside cell membrane is outside appendages, like flagella. All of them are related to the bacteria, but some are inside, some are outside. Like you have some money is with you, some money is in the bank, all is yours. Some parts are outside the bacteria, some are inside the bacteria, but all are related to bacteria and all work together for the same purpose. Archaea, we had two words, prokaryotes, eukaryotes. But we observe some microbes by microscope look like bacteria and look like prokaryotes. But after real observations, our checking, we saw it is different with prokaryotes. So archaea are different with prokaryotes. But on light microscope, they look like similar. They look like similar, but they are different. Archaea, biochemically and genetically, are as different from bacteria as you are. What does it mean? Archaea, biochemically and genetically, are as different from bacteria as you are. So it means we are different with bacteria. It means archaea are different with bacteria. So we have three groups, not two groups. Previously, suppose we have two groups, prokaryotes and eukaryotes. We have three groups, archaea, bacteria, and eukaryotes. Do you get the point? So we have three domains. We have three groups in the world. Three groups. One group, bacteria. Second group, eukaryotes. Third group, archaea. Okay? Although many books and articles still refer them to archaebacteria, because by mistake, first time they put in the bacterial groups. That term has been abandoned because they aren't bacteria. So instead of archaebacteria, we say archaea. We don't use bacteria at the end of this word. Classification of living things. This is Riyaz. We should classify all organisms. For example, at room, you classify your books. You put university books in one shelf. You put Roman books on the other shelf. You put notebooks on the other shelves. It is for simplification of the work, finding the whatever you want. So all living things divided into 3 groups. First group, bacteria. Second group, eukaryotes. Third group, archaea. Third group, archaea. So why different cars have different names? In order to know them. So if all the cars in the city have the one name, how can you differentiate them? How can you understand which one is better, which one is not better? How can you buy it? They should have different names, different groups. For example, we say plants. Why should we use plants? Because we want to know, because all plants have the same characteristics and some differences. So when we understand the common characteristics, it is not necessary many times memorize it. We say all plants have these items. Now we go through the differences. Okay? So I told you at first in the microscope, observation of archaea, yes, according to the appearance, they all look like bacteria. But recently, knowing organisms according to the appearance is not good. So how can we know them? According to the genetics, according their genetics. How much 2 organisms are related to each other and how much 2 organisms are different to each other, by use of genetics, it is phylogenetics. So what is the phylogenetics? To understand how much organisms are related to each other and how much they are different to each other, how much they are close to each other, how much they are far from each other. In animals, we know, for example, we have tiger, we have lion. How much they are similar according to the appearance, it is not a good way. By genetics, we can say how much a lion and a tiger are similar and how much is different. So according to the genetics, we can understand that archaea completely are different with bacteria by phylogenetics. By bacterial living conditions, in the same conditions, very different bacteria and very different microbes can live. So it is, for example, if we want to know one person, it is very difficult according to the place of living. For example, one person is living in the Tehran. How can we find it? According to the genetics. Without seeing one person, if any indications of genetics we have, any sample, any blood, anything, any hair we have from one person, we can find it according to the genetics. But according to the appearance, maybe we go to the another person because he is very look like to the other. Appearance is not very good clue. It is not a good way. Okay, so we have 3 domains. We have 3 domains. Eukaryotes, bacteria, and archaebacteria. But Professor Woese proposed that this word is not good because they are not bacteria. So in order to use their names, we say archaea. And we don't use the bacteria at the end of it. They are archaea. This is a phylogenetic tree. See, it is a tree with some branches. This is eukaryotes, this is archaea, and this is bacteria. They are in different branch of life. According to their genetics, they are different. We don't say they don't have any similarity. They have similarity, but they have large number of differences. For example, a lion have two eyes, a fish has two eyes. It is similarity, but lion is not look like a fish. Eukaryotes have some similarity with archaeas, but eukaryotes are not archaeas. Bacteria have some similarity with archaea, but bacteria are not archaea. Okay, they have some similarity, but this similarity is not enough to be in the same group. We have three domains, we have three groups, three completely different groups: bacteria, archaea, and eukaryotes. Instead of generation, please use the domain. We have three domain. We have three domain. Do you think which phase is more ancient? From primitive formation of the earth, do you think eukaryotes like us, like yeast, like molds, or do you think bacteria, or do you think archaea? Which phase is the first in the world, in the life? Any suggestion? Many people are writing, so a few seconds I allow to receive it. It is a question or answer? Why you bacteria? It's dependent on the nuclear. The three domain, bacteria and archaea and eukarya, all of them came with each other to the existence. Bacteria, can you repeat the question? Repeating the question. When the earth, many thousands, many million years back, it was very bad conditions in the earth. There was no air. There was not a good soil. There are very volcano. There are very bad atmosphere. 100% eukaryotes was not at that time eukaryotes, and also bacteria. Archaea are more ancient organisms in the earth. They are very powerful. Archaea, like bacteria, are microbes. They can be seen by microscope. But archaea, which created at the ancient time on the bad situation of the earth, they have very strong enzymes. They have very strong structures. Bacteria are present now, and archaea present now too. Bacteria came to the earth before bacteria, and with bacteria now both of them are present. Okay, next page. By help of those, we understood and scientists understood two domain is not correct. And archaea should come out from the prokaryotic domain and new domain added. So now we have three domain according to the phylogenetic trees of life. You can see this is one domain, bacteria. This is second domain, archaea. And this is the third domain, eukaryotes. Eukaryote, like fungi, like plants, like flagellates, trichomonads, microsporia, like animals. And we are here. We are in eukaryotes. And archaea, you can strangely, you can understand, is more similar to us than bacteria. According to the genetics, see, archaea are closer to eukaryotes than prokaryotes. Okay. Do you want 5 minutes break or continue? If no response, we continue. Okay, 5 minutes. We know that all living things in the world are in three domains. One, eukaryotes, two, prokaryotes, and three, archaea. We want to see some common parts we are discussing. First, cell membrane. I told you we cannot find any cell without cell membrane. It is very essential. See, cell membrane of bacteria and eukaryotes, both are ester-linked lipids. Ester-linked lipids. So our cell membrane is look like to cell membrane of bacteria. That cell membrane of archaea is different. It is ester-linked lipids. Ester-linked lipids. Ester and ester is very simple. See, this is glycerol. In archaea, there is ester linkage. In bacteria and eukaryotes, it is ester linkage. Ester linkage. So our cell membrane is look like cell membrane of bacteria. But cell membrane of archaea are different to all others. It is different to all others. So first one, ester-linked lipids in bacteria, ester-linked lipids in eukaryotes, but in archaea it is ester-linked lipids. Outside the cell membrane in bacteria is cell wall. Its composition is peptidoglycan. It's a composition. Peptidoglycan. It has two parts. Peptido means one constituent is peptide, amino acids, and glycan. Glycans means carbohydrates. It is a combination of amino acids and carbohydrates. So, cell wall of bacteria is peptidoglycan. But some eukaryotes doesn't have cell wall, and some like algae, some like fungi, cell wall composition is completely different. Completely different. Archaea has pseudopeptidoglycan. Peptido means amino acids. Glycan means carbohydrates, but different with peptidoglycan of bacteria. So we say pseudopeptidoglycan. Look like, but different. Look like and different. So it is pseudopeptidoglycan. Gene structure, archaea and bacteria, one circular chromosome. Each bacteria, one circular chromosome. Similar translation and transcription to eukaryotes in archaea. So genome works in archaea is more similar to eukaryotes. And here you say multiple chromosomes in eukaryotes and translation and transcription similar to archaea. So their works, archaea and eukaryotes, are similar. Unique translation and transcription. Translation and transcription almost is different with the others. The other part, internal cell structure. Internal the cell, there is no membrane. There is no nuclear membrane in archaea like bacteria. So this is similarity between bacteria and archaea. No membrane bound organelles or no nuclear membrane. They have nucleoid. But in eukaryotes, membrane bound organelles and nucleus. Nucleus and organelles all are nuclear bonded. All are nuclear bonded. Metabolism. Catabolism and anabolism. Metabolism. Different types in archaea. For example, like methanogenesis. They get carbon dioxide and produce methane. Unique to archaea. Some of this metabolism are unique to archaea. It won't happen anywhere in another organisms. In bacteria is also various. Some bacteria are photosynthetically, some aerobic, some anaerobic respiration, some fermentation, and some autotrophs. Okay, one by one, we check it. What is the autotroph? Some bacteria like algae, some bacteria like plants can capture the sunlight and put this energy in the organic matters. They can get the energy of light. They are photoautotroph. Some have respiration. What was the respiration? Use food and produce energy. Use food and produce energy. If it is depending on the presence of oxygen, it is aerobic respiration. If it is done without oxygen, it is anaerobic respiration. So what is the respiration? In the next week, I explained completely. But right now, what is the respiration? It is a tool to use food and produce energy. If it is by use of oxygen, it is aerobic respiration. If it cannot be used oxygen and use another things like nitrates, like phosphates in a state of oxygen, it is anaerobic respiration. Do you think anaerobic respiration is more similar to aerobic respiration or to fermentation? Anaerobic respiration. Do you think it is more similar to aerobic respiration or fermentation in general in the products? Okay. Another response? Okay, see, aerobic respiration and anaerobic respiration completely are the same with one exception. So aerobic respiration and anaerobic respiration completely are the same way and same result. One exception. First is by the use of oxygen. Second is without the use of oxygen. All the direction, all the way are the same. And they produce 36 ATP. 36 ATP. But in fermentation, only the first part of the way happening and only produce 2 ATP. And only ATP production is 2. We as a human, which one we have? Aerobic respiration or anaerobic respiration or fermentation or autotrophy? What do we have? Aerobic respiration or anaerobic respiration or fermentation or autotrophy? Who can answer? We have some pioneers. For all questions, a few of them are volunteers all the time. I am waiting for them as well as the others. Did you get the question? Did you receive my question and understand it? We are human, okay? I'm saying we as a human, do we have aerobic respiration or anaerobic respiration or fermentation or autotroph ourselves? Which one do we have? Aerobic respiration, of course. See, why do we go to shop to get food? Why do we go to the restaurant? We want energy. When we get food, it absorbs, it goes to the cells, it goes through the glycolysis, Krebs cycle, and then go to the mitochondria to produce ATP. The final acceptor of electron is oxygen. If there is no oxygen, there is no respiration. If there is no respiration, there is no metabolism. If there is no metabolism, there is no life. So why do we eat food? We eat food because we want energy. And this energy in the respiration needs oxygen. So we have inhalation. We get oxygen to our lungs to send it to all cells for aerobic respiration. Do we have fermentation? Anybody? Do we have fermentation? Do you have any idea? When and where? Mrs. Aya Qazi, dear student. Okay, if so, when and where? Okay, any more? See, when you go to gym and you go to a sport, when you want to have a pleasant sport, what is the guide say you? It say you do it aerobic exercise. Aerobic exercise means as much as do exercise which all your cells get complete oxygen. But if you run very fast for 1 kilometer, what's happening? Why your foot muscles will be painful? When you go very fast, not sufficient oxygen reach to your muscle cells. So what happened? So cells need energy because you are running and there is not sufficient oxygen. So fermentation happens and lactic acid production happens. And a few days you have foot pain because lactic acid. And after some days, sometimes, and remove this lactic acid, again you go to the healthy way. But if you go through the aerobic exercise, after finished the exercise, there is no tiredness. There is no tiredness. Our body is not for fermentation. Its construction is for aerobic respiration. What's about anaerobic respiration? No, we don't have anaerobic respiration. We cannot use anything else like nitrate or phosphate instead of oxygen. No, we don't have anaerobic respiration. But during hard working, during hard exercise, we have ourselves go by force to the fermentation. Okay, and this is in eukaryotes. Some eukaryotes have photosynthesis, like what? Like plants, like algae. Some bacteria has photosynthesis. Like what? Which bacteria has photosynthesis? Like cyanobacteria. It is a photosynthetic bacteria, cyanobacteria. But eukaryote, algae. The last step, reproduction. Reproduction. Very good. Excellent. Archaea and bacteria both have asexual reproduction. They don't produce gametes. They have asexual reproduction. All the organelles and chromosome will be double and each separated to each side. We have two. The reproduction of bacteria and archaea are logarithmic. See, 1 organism, 2, 4, 8, 16, 32, 64, and so on. So this is asexual reproduction and logarithmic reproduction. All eukaryotes have asexual reproduction. Algae, plants, and the others. But most of them, besides asexual, they have sexual reproduction too. So all organisms, all living things has asexual reproduction. For example, one cells of my hands die. So the new one will come by asexual reproduction. Asexual reproduction. In molds, in fungi, and the others, there are many types of asexual reproduction, but in some cases, in some microbes, in some eukaryotes, like the agent of malaria. What is the agent of malaria name? What is the causative agent of malaria? Anybody tell me? Very good. Very good. Dear student, Sauda. And the others, 100%, many of you knows, Plasmodium. It has asexual as well as sexual reproduction. Where happens the sexual reproduction of Plasmodium? You know, it has two hosts, human and anophel. Sexual reproduction happens in human or in anophel? I'm waiting. Sexual reproduction of Plasmodium, a eukaryote, happens in human or in anophel? Many volunteers are writing. I'm happy. Yes, I told you it has sexual reproduction as well as asexual reproduction. And my question is that sexual reproduction happening in human or in anophel? The others are writing. It happened in host. Okay, it has two hosts. One host is a human and the other host is the anophel. It has two hosts. Unlike you wrote and never you forget it, sexual reproduction in happening in anophel, and in human only asexual reproduction happening. Only asexual reproduction in human happening. Okay, by the way, this is this page. I explained this one. Some answers are coming by delay. My answer was, I told the answer. The sexual reproduction of Plasmodium happens in the anophel as the vector, and the asexual reproduction happens in human. Okay, again we return back to the bacteria. Physical and chemical properties. Physical and chemical properties. Chemical composition. What is the chemical composition of a bacteria? Water should be say at the first. All the things are alive with water. Water is the most important thing in the world. Water is more important than gold. Water is more important than oil. Water is more important than anything else. Inorganic salts. some inorganics, proteins, carbohydrates, lipids, nucleic acids. Here are some chemical compositions of cell, of bacterial cell. This is Aye Ghazi, dear student. Is important? I don't know. It is answer or question. What is important? I don't know. You can repeat it. Some requirements, some foods, yes, of course. Okay. Okay, Mr. Hussein Sabah is tired. What should we do now? So I give you 5 minutes break to be refreshed. And after 10-15 minutes, you go to the rest. But I don't think so. I think you don't go to the rest. You go to some other works. By the way, a few minutes waiting. We are very close to finish, and a little the previous group with more expedite. They are on the next slides. Now we are back side of the previous group. A few minutes have a cooperation. It will be finished very soon. Okay. Bacteria needs foods. Some requirements are in macronutrients or macro elements. But some requirements are in the micronutrients, trace elements. What micronutrients? Like carbon sources for energy production, for using in the building of the cell. Oxygen. For those which require oxygen, it is macronutrient. But oxygen for some bacteria is toxic. They don't like it. So oxygen is not necessary for all bacteria. Some bacteria need it. Some bacteria, if it is present, very good. Not present, go another way. Some organisms, if oxygen present, is very toxic and they will be killed. Hydrogen, nitrogen source for construction of amino acids, for proteins, for enzymes. They need it in large quantity. Sulfur, phosphorus, and other metal ions like potassium, calcium, magnesium, and iron. The first, carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphate are components of carbohydrates, lipids, proteins, and nucleic acids. Now you understand, now you know why they need in large quantity, because all the body of a cell is carbohydrate, lipid, protein, nucleic acid, and for duplication, for multiplication, so bacteria should get carbon, oxygen, hydrogen, nitrogen, sulfur, and phosphor as much as possible. And a little they need micronutrients. They need a little micronutrients. For growth, only nutrients are not sufficient. Nutrients is okay. It is necessary. But environmental pH, environmental temperature, environmental oxygen, and osmotic pressure. For example, one bacteria live in Oman Sea. So the amount of salt in the water is high. We cannot get that bacteria and put it on the spring or in fresh water. It cannot live because osmotic pressure is completely different. Osmotic pressure. So nutrients, which nutrients bacteria need? Carbon source for breaking to produce energy, for use them to construct the building of the cell. Nitrogen source for amino acids for protein synthesis. Inorganic salts, trace elements for different enzymes, different parts, they are needed. Growth factors, some bacteria, whatever a bacteria need for growth is growth factor. Bacteria excrete extra growth factor outside to be used by the others. Water is a nutrient, is very necessary and important nutrients. If one bacteria can use the simplest form of carbon, like carbon dioxide, they can use inorganic molecules to produce organic molecules. They are autotrophs. If any bacteria or any microorganisms like us cannot use the simplest inorganic molecules to produce organic ones, they are heterotrophs. Heterotrophs attack to living cells, they are parasites. If heterotrophs attack to the dead tissues, for example, one broken leaf or trees or a dead particle of organism things, we say they are saprophyte. So parasite attack to living thing. Suppose one fungi attack to the leaves of a tree. It is a parasite. When the fruit is on the tree,